He's a professor of medical genetics at UBC. He's a clinical geneticist who holds an MD degree from Tulane University and a PhD in genetics from the University of Washington. His research has led to a lot of advances, especially in neurofibromatosis type 1, which he's an international expert. And he's helped to enumerate the complex and variable spectrum of the clinical features, including formulating recommendations for genetics and clinical diagnosis, and as a result has enhanced the patient care in patients with this condition. His key research discoveries of novel molecular and cellular hallmarks that distinguish types of neurofibromas, which are benign tumors that are in NF1, have provided new insights into the pathogenesis and progression of that condition, NF. One of the reasons why we invited him today, however, is that over the past decade, he's been a pioneer in the development and applications of cytogenetic and genomic tools to help understand the genetic causes and clinical consequences and to improve the diagnosis of intellectual disability syndromes. And so he has a lot of experience in microarray and exome technology. He's also a recognized expert in teratology and recently has focused on determining the risk for birth defects from women using antidepressants during pregnancy. He's won numerous awards for teaching and research, participated in many committees and advisory groups at UBC and externally, and published over 230 peer-reviewed papers, articles, chapters, and has given over 50 invited presentations. So I introduce you once again to Dr. Jan Friedman.
Thank you, Sarah. It's my great pleasure to be here and to tell you about some of the things that we, and it definitely is many people in Vancouver, the work I'm going to tell you about, have been involved in for the last few years, looking for mutations that cause Mendelian disease using next-generation sequencing technology. So historically, the way that we were able to identify causative genes has required rare characteristic phenotypes with little or no genetic heterogeneity and either large Mendelian families or chromosomal abnormalities. Those are really the only two ways we've been able to find genes that cause disease. So the large Mendelian families, the approach has been linkage, as shown here. And this was, for example, how the gene that causes Huntington's disease was originally found 30 years ago, almost 30 years ago now. Chromosomal abnormalities are the other major way it was found. An example here is with the deletion of 20p that was seen in a few children with arteriohepatic dysplasia. Most kids with arteriohepatic dysplasia did not have this chromosomal abnormality, but a handful of children did. And when the deletions that they had were overlapped, it was found that there was only one gene, JAG1, that was common to many of them, and that turns out to be the gene for arteriohepatic dysplasia. So this is the second way that many disease genes were found. And I remind you of these only because these technologies, these approaches are still useful. But now we're combining them with next generation sequencing. Can I turn off this light without turning off all the lights? Is that possible? So the next generation technology, sequencing technology, I think you all know about. In comparison to the sequencers that were used for the Human Genome Project, you get more than 200,000 times more sequence per run, and you can do an entire human genome in about a week now instead of 13 years. So these are huge, huge technical advantages, but the biggest advantage of all is in the cost, which has fallen dramatically, much faster than Moore's Law, which is the doubling of the computational power on a computer chip every 18 months. So this huge reduction in the cost of sequencing produced by next generation sequencing has really revolutionized what we've been able to learn about the genome. And what have we learned? Well, we've learned about the unity of life. We've learned that the proportion of human genes represented in other species is incredible. So 79% of our genes are present in mice, 76% in dogs, 73% in horses. Half of our genes are present in chickens, and almost 60% in zebrafish. So the fact that the genes that we have are pretty much the same as the genes that other species has came as a surprise to many people who thought that humans had to be very different. In fact, we're very similar. We've also learned that we are a lot alike. That any two people share about 99.5% of their genomes. So if you look at the person next to you, 99.5% of your genome is identical to the person sitting next to you. But we've also learned that there's no normal human genome. The other side of this is that any two people differ by about 0.5% of their genomes. So two people who are completely healthy, normal, have differences in their genomes in about 5 out of 1,000 of every base pairs. When I was a graduate student, I was taught that there was a normal genome, normal genes, we called that the wild type, and anything that varied from the wild type was abnormal. Well, we now know there is no wild type. There is no normal genome. There are many different flavors of normal genes in normal people. And this immense genetic variation we see when we look at people's faces, and we see in spades when we look at their genomes. So any two people, their genes differ by about 30 million base pairs. Every person has about 6 billion base pairs, and when we try to interpret the data from their genome, we have to sort out the 6 billion base pairs. We have to sort out the 30 million differences to try to find the 1 or 2 or few base pairs that may account for a disease. That's the problem that we're faced with. Moreover, as we've done a number of normal genomes, we found that everybody carries damaging variants. Things that we would have called mutations are present in every normal person. Hundreds of these. So the key challenge we face when we're trying to do exomes or whole genomes to try to find disease genes is to interpret these variants. Here's an example of the most well-sequenced person, actually, cell line that exists. This is an NIH, or a Ceph cell line that has been studied by every possible sequencing technology many, many different times. It's the standard that people use. And in this cell line, there are 79 now validated heterozygous mutations that we would predict would cause disease. Stop site, splice site, frame shift, indels, large deletions. And there are 18 of these that are homozygous within this cell line. But this was from a normal person. So when we get exome sequencing data, we always, or the bioinformaticians, always select the variants for us. They don't give us 30 million variants. They give us a list of usually a few hundred. And these are selected by quality of the sequencing, by the extent of the coverage, so for technical reasons, by the rarity. Usually we get rid of things that we know are polymorphisms in the population. But these alone are not enough. This doesn't give us what's pathogenic. In order to find the pathogenic gene, in order to find Mendelian disease genes, you got to have a gimmick. There has to be more to it. You got to have some clue to find the right gene or you'll never find it. So how do we prioritize these variants? We prioritize them by what we know, what we've known for years. So inheritance of the disorder. If we're looking for a dominant, it should segregate properly in the family. If we're looking for a recessive, one abnormal allele, you got two abnormal alleles at the same locus, and one of those abnormal alleles should come from each parent. The uniqueness of the phenotype can be a big help. Again, if you have a syndrome that only occurs in the context of mutations of a certain gene, that really helps. Availability of other affected individuals with the same syndrome or other family members, whether they're affected or unaffected, helps a lot. And genetic heterogeneity hurts. So if you're looking for a genetically heterogeneous condition, it's a lot harder than if you're looking for a condition that can only be caused by mutations of a single locus. And there are several strategies that people use, and we've used, to find mutations and that cause disease. And we start off with exactly the same way we started off finding genes before we had next generation sequencing, with linkage. So linkage in families is a good way to narrow the search space. Whether you're talking about dominant or recessive genes, you want to find variants that segregate with the disease, or alternatively, exclude variants that do not in multiple family members. And here's an example of finding a gene this way. So this is a condition with neurodegeneration. This is a family of Christina Diaz's when she was in Vancouver. The proband was normal at birth, and in infancy, at 18 months, she began, her weight gain slowed down. She didn't actually lose weight. At 22 months, she had a mild acute gastroenteritis and developed ataxia and dysarthria. And an MRI showed white matter abnormality. Subsequent neurodegeneration with remissions and relapses, so over the next 3 years, she got better, got worse, got better, got worse, progressively worse. And at 5 years of age, she died with cardiac failure and cardiac hypertrophy. Her older sister was normal at birth, had mild developmental delay and mild hearing loss, but otherwise was completely healthy. At 5 years old, she got chickenpox, developed a seizure, had progressively deteriorating mental status, was found to have white matter changes on the MRI, had a progressive course with remissions and relapses, and died 19 months later. So she actually died just a little bit before the younger sister was born. And this is the pedigree. This was a Lebanese family that was extensively inbred. The coefficient of inbreeding, the parents of these children were consanguineous, but there were multiple loops of consanguinity within the family. The coefficient of inbreeding was equivalent to these kids being the offspring of half-sibs. So we were looking for an autosomal recessive, and we used linkage. And we had normal sibs, and we had normal parents. So we did whole exome sequencing on all 5 sibs and both parents. The analysis was for a homozygous mutation that was present in both affected sibs, but not in the unaffected sibs, and not in either parent. There are a lot of things that were homozygous in the parents too, because they also were the offspring of consanguineous babies. The gene that was found was porphyrin 1, and a mutation was found, this specific mutation that we've described here, is identical to a mutation that had previously been reported in a completely different genetic disease, familial hemophagocytic lymphohistiocytosis, or FHL. These kids had no features of this other disease, except they did have defective innate immunity. And it turns out that their neurodegeneration reflected that abnormal immunity to two different viruses, started off with two different viruses. In retrospect, when we look at FHL patients, they may have neurodegeneration. So neurodegeneration of this kind can be an occasional feature of familial hemophagocytic lymphohistiocytosis. And this is almost certainly the right gene. But completely different presentation of this disease in these kids. The other strategy that was used in this family is a homozygosity strategy, which you can use in consanguineous families like this for autosomal recessives. And the idea is to select variants that lie within the large regions of homozygosity. So SNP arrays were actually done in the family. The area of homozygosities were identified on the SNP arrays, and that's where we focused the search. We actually had to extend the search beyond the biggest areas of homozygosity, because these were actually this gene was actually in a somewhat smaller area, but it still was in an area of homozygosity. So here's another example of this strategy. This is a 4-year-old girl who I saw about 15 years ago with microcephaly and profound intellectual disability. We did a karyotype that was normal, we did a brain CT that was normal, we did plasma amino acids that were normal. It was first cousin mating again with additional loops of consanguinity. There was a history that the brother had died of pneumonia at age 7 and had been similarly affected with microcephaly and profound intellectual disability, but this child died before the family came to Canada, and there was no other information on him. We counseled him. There was a 1 in 4 recurrence risk. We couldn't do prenatal diagnosis because we didn't know what the cause was, and the mother was, of course, pregnant at the time. My colleague, Anna Lehman, saw this family again just recently. This is the child who was born of that pregnancy. She was I guess it was 18 years later, because she was now 18 years old. She has microcephaly, profound intellectual disability. Brain MRI, chromosomal microarray, metabolic screen were all normal in this child. Her older sister, who is the one that I saw, is now 23 with microcephaly, profound intellectual disability. Anna did SNP array and found homozygosity studies that reduced the region of likely homozygosity where this mutation probably lies to about 256 megabases, about 8.5% of the genome. She then did exome sequencing in the older sister and found 9 genes with homozygous rare variants. And of these, 1 gene was known to be associated with the genotype. In SUN2, a truncating mutation. This is a recognized intellectual disability gene that is associated with microcephaly too. So almost certainly the right gene in this family. So another strategy that can be used for recessive diseases, called the double hit strategy. This is for families where there's not consanguinity. And in this case, we select for variants that are compound heterozygous, different mutations of the same locus. Or else it could be homozygous, but more likely, if the parents are unrelated, compound heterozygotes. Here's an example of this. This is a family of Isabel Filges. Isabel's a postdoc in the lab. This was a healthy, non-consanguineous couple who was referred for genetic evaluation in their second pregnancy because of recurrent multiple fetal anomalies. First pregnancy, at 21 and 4/7 weeks, the ultrasound showed fetal growth retardation, severe microcephaly, cerebellar hypoplasia, and bilateral renal agenesis. Pregnancy was terminated. Female fetus, findings were confirmed. Counseled possible autosomal recessive, recurrence risk, maybe as high as 25%. We weren't sure. Chromosome analysis was normal. Second pregnancy, 18 and 5/7 weeks. Ultrasound showed fetal growth retardation, severe microcephaly, aerodencephaly, cerebellar hypoplasia, bilateral renal cystic dysplasia, and hypoplasia. A little different, but almost certainly the same condition. Pregnancy was terminated. Female fetus, findings confirmed. Cytogenetic analysis was done on CVS in both cases, was normal. Chromosomal microarray was done in the second fetus, no pathogenic findings. Pattern was thought to resemble a ciliopathy, probably autosomal recessive, but didn't exactly fit any known syndrome. Parents subsequently had a normal daughter who's now 2 years old, so there's a happier outcome here. Isabel did whole exome sequencing on frozen CDS from the second pregnancy and blood from both parents. She postulated there was a compound heterozygote for inactivating mutations of a ciliopathy gene, and so she put together a list of genes that are known as suspected to be involved with function of the cilia. There were 1,644 such genes. And she looked for one of those being compound heterozygous. And she found 35 loci where there were rare compound non-synonymous variants in the child that were inherited from the parents. There were 3 loci of these 35 that were on her ciliopathy gene list. And there was one locus, a member of the Kinesin family, where both variants were truncating, and it showed the expected segregation family in the unaffected girl as well as in the affected parents, all of whom were heterozygous. It turns out that a spontaneous mutation of the mouse exists for this particular gene. And in that mouse model, there's growth restriction, microcephaly, and cerebellar hypoplasia, as well as motor impairment. Pretty good match for a mouse model. And the mouse model was proven to be pathogenic. This was a spontaneous mutation. But the mouse model, it was proven that they had the right gene by doing a knockout of the same gene and showing the same phenotype. So the mouse model definitely is the right gene. We think it's the right gene in the mouse. And Isabel has now sent this paper in for publication. This is an example also of a candidate strategy where you prepare a list of suspected candidates based on the presumed function of the gene. And instead of having to look through 20,000 genes, you look through a smaller number. In this case, it was still 1,000. But often it can be a smaller number than that. You select these variants by predicted gene function, and you can use this with either dominant or recessive models. So the other genetic family history model that we use is based on the fact that in Mendelian disease, we know that mutations are rare. They occur in about 1.5 times 10 to the -8 generations per mutations per base pair per generation. But new mutations are common in people with Mendelian dominant diseases that reduce fertility, because we wouldn't see those diseases if there weren't new mutations. And it turns out that new mutations are common because we have 6 billion base pairs that are potentially mutatable. So any one of us carries 70 to 110 or so, about 100 mutations that are not present in either of our parents. Most of these are out in DNA that doesn't have any function that we recognize, or in exons that change function that aren't critical, or aren't critical in a heterozygous fashion. But we all carry these mutations. So, when we see this kind of a family history, which we see all the time in the genetics clinics, this is the most frequent family history we see, that is a negative family history, we don't know whether we're dealing with a condition that is not genetic, or an autosomal recessive, or an X-linked recessive. In small families, we very frequently see this, or whether we're dealing with a dominant that's a new mutation. And the dominants that are a new mutation provide another way that we can find disease genes, because we look for mutations that are present in the child that are not present in either parent. Another approach is the overlap strategy, and this is one that's often used with the new mutations. These are when we have basically a syndrome. We have a rare phenotype. We have unrelated probands who have the same very rare, unique phenotype, but the two kids have exactly the same thing. Could be dominant, could be recessive, either is sporadic, usually is sporadic, occasionally they're small families. But this one depends crucially on good genotyping. You've got to know that the kids have the same syndrome. Example of this that Bill Gibson recently published is with the Weaver syndrome. This is a condition, as you know, that's associated with intellectual disability and generalized overgrowth, marked enlargement of the head, characteristic facial features, very rare syndrome. Only about 40 cases have been reported in 35 years since it was first described. It's usually sporadic, but there's one report of parent-to-child transmission suggesting it is a dominant, and suggesting that the sporadic cases are new mutations. These are the facial features. The child on your left is the first child that David Weaver reported with this condition. Child on the right is Bill Gibson's patient. He sequenced the exomes of these two patients, found 24,000 variants in one and 25,000 variants in the other. There were 17 novel de novo variants found in one patient, 16 in the other, but only one was common to the two children, and that one was EZH2, a novel de novo variant in both patients. A third patient that David Chidiat provided was then sequenced just for this gene, and another variant, de novo variant, was found. So this is certainly the mutation that causes this condition. So these are the gimmicks that you can use to find these mutations. But how do you prove that you've got the right gene? This is the problem that drives my graduate students and postdocs absolutely crazy, because you've got to be sure. And there's a few things that we do. The first thing you always have to do is technical validation. Usually we do that with Sanger sequencing because mistakes are made. Next generation sequencing is pretty damn good. It's 99.9+% accurate, but you're looking at 6 billion base pairs if you're looking at the whole genome. or you're looking at 2 to 3% of that if you're looking at an exome. So there's still lots of chances for error. And you've always got to do Sanger sequencing to validate. You have to do the genetic validation in the family. Depends on the pattern of inheritance, but if it's recessive, you should find one mutation in each parent. Unaffected siblings should not have both mutations. If it's a new dominant, you shouldn't find the mutation in either parent. Whatever the pattern is, you should validate that in the family. Clinical validation in unrelated individuals. This is this overlap strategy, looking at other people who have the same mutation is very powerful, not always possible. When it's not possible, we have to do functional validation, either in patients, and you can do this, for example, if you're looking at a biochemical disorder, you may find a way to functionally test the enzyme, for example. Or in model systems, which may be cellular systems or an animal model. But it's important to know that because the locus is conserved and because the nucleotide that you're looking at is conserved, that's not enough to prove that it's pathogenic. This can occur in variants that are not pathogenic, in polymorphisms. It's important to know that expression in the right tissue is not the same thing as pathogenic. And that's true in spades if you're talking about the brain. Almost every gene is expressed in the brain at some point in development. So it should be expressed in the right tissue, but finding it expressed in the right tissue isn't enough to prove it's pathogenic. The fact that it's a rare variant isn't enough to prove it's pathogenic. We all have lots of variants that aren't in dbSNP. It's good for it to be a rare variant, but it's not enough to prove that it's pathogenic. Predicting that it damages function to the protein is not enough to prove that it's pathogenic. There are lots of good programs out there that can predict damage. Any of you that have tried to use these know that they don't all agree, that they give you a different kind of prediction. But in any case, even if they do all agree, it's not enough to prove that it's pathogenic because there are lots of polymorphisms that are predicted to damage protein function. And most important for our graduate students and postdocs is a good story doesn't mean it's pathogenic. A smart graduate student or postdoc can make up a good story for any gene, for any disease, I'm convinced. But that's not enough to prove that it's pathogenic. The problems that we have are the old problems of genetics, the principles we have to go back to, pleiotropy. More than one phenotype may be associated with different or identical mutations of one gene. And the porphyrin story is an example of that. You have a completely different phenotype, same mutation of the same gene. And locus heterogeneity. Mutations of more than one gene can produce the same phenotype. And we know now that this is the rule. It's not the exception. There are few diseases, clinically defined diseases, that have a single unitary locus that they're associated with mutations of. Usually there's more than one, sometimes there's hundreds. Or for ciliopathies, thousands. So when we prove pathogenicity, my mother said, don't believe everything you read. When we're trying to prove pathogenicity, don't believe anything your computer reads. We know that sequencing errors are common, especially at read depths less than 10 for a particular mutation, and we often encounter read depths of less than 10 if you're going an average or a median depth of 30 in a an exon. Misalignment is frequent. The alignment algorithms don't always work. They get confused by pseudogenes, and they get confused by indels. And HDMD doesn't always work. Probably half or so of the so-called mutations in the human genome mutation, gene mutation database are not true mutations. Most of them were put in before we recognized the frequency of polymorphisms that can be predicted to inactivate genes or alter genes. Most of them used as comparison a single, say, 100 normal individuals, usually Caucasian, and then they opened the lab to test in all kinds of patients, many of whom are from different populations and may have different variants that may occur in that gene and be interpreted as rare variants, even though they're common in the population they come from. So probably about half of the mutations in HGMD are not true mutations. So when it comes to proving pathogenicity for a clinician, nothing beats a syndrome. Good phenotyping is absolutely essential. Family history is absolutely essential. The rule of three cases with the same mutation, same phenotype, you probably got the right gene, still works. Don't forget your basics. They're still important. When proving pathogenicity, the cases I've talked about are the low-hanging fruit. Mendelian disease is the easiest. If you're looking for mosaicism, it's way harder. If you're looking for digenics, it's way harder. If you're looking for multifactorials, it's much harder. Okay, so the sequencing I've talked about is research. I want to talk just a second about clinical sequencing. And to draw the distinguish between research sequencing, which is done to find genes and to elicit genetic mechanisms, and clinical sequencing, whole exome or whole genome, which is used to demonstrate pathogenic mutations in a Mendelian disease, to demonstrate mutations that we know causes the disease. Clinical sequencing is not research. It's intended to find a known mutation that causes a known disease. We usually use exomes because analysis of whole genome data is difficult and time-consuming, and the cost of analysis is much greater than the cost of sequencing. We know that about 85%, or it's estimated that about 85% of known disease mutations occur in the protein coding portions of genes, which are only 1 to 2% of the genome. This is probably an overestimate because we've been looking under the streetlight, but most of the disease mutations are in the exome. Genomic selection methods can be used to enrich for that exome. When we look at an exome, we're looking at about 20,000 genetic loci, about 180,000 exons, about 50 megabases instead of 6 gigabases of the genome, about 1 to 2% of the genome. It's cheaper, and we can interpret it much better. But it's a transitional technology. Don't fall in love with it. It's going to be replaced by whole genome sequencing in the near future as the costs continue to fall and the ability to analyze whole genome data improve. We know that exome sequencing is incomplete. We know we don't get all coding regions. We know we don't get any non-coding regions, and so we don't get all mutations with the exome. Whole genome sequencing is more complete. You also get more uniform coverage. One of the problems with exon capture is that you capture some exons better than other exons and some not at all. The ENCODE data indicates that at least 20% of the genome is functional. The exome is only 1 to 2% of the genome. And the algorithms to detect copy number variants and other structural variants are more robust with whole genome sequencing. So we may be able to do away with microarray when we go to whole genome. We probably will be able to. So whole genome sequencing can also detect copy number variants and structural variants that are below the resolution of chromosomal microarray. They can go down to deletions that are a few base pairs. De novo assembly with whole genome sequencing has allowed the detection of balanced structural rearrangements, which you can't do with exome sequencing. And also there are techniques to find trinucleotide repeat expansions now, which you can't do with exomes. Cost reductions are likely to be greater with whole genome sequencing, so it's converging with whole exome sequencing. The price of sequencing itself has fallen much more than the price of exome capture. So the two are getting closer together in terms of costs. So why not go to whole genome sequencing today? Why not put this in the clinic today? There still are problems with interpreting the data. It's hard. The genetic counseling for whole genome sequencing is hard. The model that we're using, which is based on things like BRCA1 and 2 or Huntington's disease or cystic fibrosis, they're probably the wrong models. They don't apply one gene at a time, completely different than when you're looking at 20,000 genes at once. Incidental findings you can't ignore. You've got to pay attention to them. And the cost of the sequencing itself is still non-trivial, but it's probably the lowest thing on this list now that's preventing this from coming to the clinic. But the other thing is inertia. Inertia on our part as clinicians, inertia on the laboratory's part, inertia at our ministries of health. They're not used to doing things this way. So, I'd be glad to take questions if you have them. Can we turn the lights back up? Yes?
A couple questions. For things where you can get pretty good linkage in incompletely dominant disorders, we found those very hard. Have you the same experience in your studies?
Where you have incomplete penetrance? Yeah, it's a lot harder with incomplete penetrance. It's a lot harder if you have variable expression. This is exactly the same thing that was true before we had next generation sequencing, and next generation sequencing doesn't solve the problem. The best solution to that we found are really big families. If you have lots of families and you test people that are far from each other, but you know they carry the same gene, then you have lots of power. You can narrow your search space a lot. But if you just have small families, it makes it a lot harder. I don't have a magic solution to that.
In many of these projects, it seems quite clear that the variant is not exonic but rather regulatory. Have you got any strategies for dealing with those?
Well, whole genome sequencing. We're dealing with some of those ourselves. We have one particular disease that we've been working on for a year. It's a very rare and very unique phenotype. We've done, I think, 8 exomes at very high depth, looking for mosaicism. It's just not there. We know it's X-linked. We even know what chromosome it's on. It's not in the exome. So we're doing whole genome, and we're looking. But the tools for looking in the outside the exome are not very good. They're better. We've got the ENCODE data now. NIH just put out a request for proposals for software to interpret non-exonic mutations, to identify non-exonic mutations. So hopefully somebody will develop even better stuff, but it's also a very difficult one. We know there are mutations there.
So just, people have talked about whole genome sequencing of newborn screening blotters, or, if there's capabilities of even doing that on the fetus. People are talking about having people that from birth, having all this information about them. What are your thoughts about the next 10 years? Where do you think things are going to be heading?
Well, I think technically that will be possible, but it gives me the willies. Technically, I think it also will be possible to do that prenatally from the mother's blood. It's just a question of sequencing to more depth than we're doing now. Individual genetic mutations have been identified in cell-free DNA in the mother's blood, so there's no reason to think. And chromosome-wide coverage has been generated to detect trisomy in the mother's blood, so there's no reason to think that we won't be able to get the whole sequence, and you'll be able to do it from blood spots as well in the newborn. But everybody's going to have mutations. Everybody's going to have variants of uncertain significance. Many of these are going to be in diseases that we can't do anything about to prevent. But finding that information may at times be helpful. For example, knowing pharmacogenomic variants ahead of time may help you choose the right anticonvulsant or the right anticoagulant for a patient. But it may also preclude them from getting life insurance. It may also prevent them from getting certain jobs. And so I think it's very troubling. We don't know how to deal with that, and I don't think we're ready to be using this technology routinely on everybody as a birthday present. Yes?
So you mentioned one of the barriers was interpretation of data. I'm not a clinician or a scientist, but just tracking some news stories that have cropped up in Nature and other journals lately, talking about sort of the growth of bioinformatics firms that are providing those services. So effectively, healthcare providers outsource that step to some extent, or at least there's a business model that will cater to that practice. In a Canadian context, my understanding is a lot of those new businesses are in Silicon Valley, et cetera.
Yeah.
Can you think through, or does that present concerns or advantages in a Canadian setting if you think about that kind of business model?
You mean setting up such a business in Canada? It is being discussed. I don't know. So there is no solution to this, and none of those companies have the answer yet. I think we would all, clinicians certainly would all benefit by much better software. The analysis that we do on these exomes now, it takes a hell of a lot of person hours. Even when we work mostly with the Genome Sciences Center in Vancouver. There's about 80 bioinformaticians there, and they have a pipeline for analyzing this, and they give us a very well annotated list of variants, but there are still 100 variants on that list that the clinician has to go through and figure out what makes sense and what doesn't. And although there is software that allows you to look at each variant in the context of the annotations that exist in the various genome browsers, you still have to go back to the papers and figure out what's going on and interpret it. And we're a long way from solving those problems. I think it'll come. It's come with chromosomal microarrays, much better than it used to be. It's not trivial, but it's much better than it used to be, and I think it will be. So whether that will come from selfless academics who make this software available free, or more likely probably from companies, and we'll have to buy it, I don't know. I don't think Canadian companies are at a particular advantage or disadvantage for this, because no matter where it's produced, if you've got a really good product, people are going to be using it because we desperately need it. So I think it's not an unreasonable thing to go after if you want to start that business. Yes.
I have a trivial question, but as a clinical geneticist, are people coming to you prognostically with their 23andMe results in hand and asking you to interpret those?
Yes. More often to our genetic counselors who hate it. But yes, they are coming. And we're not very well prepared for it. We don't train people very well to deal with that. And actually, 23andMe is not the biggest problem, because 23andMe has a terrific website that has lots and lots of information on it, and about all of their variants, and links to the papers, and at least you can refer the patients back to that website. It's more of a problem with some of the others that don't have such good information, but it's definitely happening. We're also having, I had a neurologist consult us not long ago because a patient brought in some pharmacogenetic testing that he had done for he has a seizure disorder, and wanted to know if they should change his anticonvulsants, which he'd been on for years and seemed to be working pretty well as far as the doctor was concerned. So it's coming in many different areas, and certainly people who have rare diseases and don't have a diagnosis are coming asking for exome sequencing or whole genome sequencing. Yeah.
And in the course of your research and your exomes and so forth, have you found yourself in a position where you've had to return incidental findings to your participants?
Yes, we have. We actually first encountered that in some of our microarray studies, chromosomal microarray studies. For example, we had a child with an intellectual disability doing trios, and the mother was found to have 3 X chromosomes. We had another child, little girl with intellectual disability, who was found to have a deletion of the entire BRCA2 gene. So these things do come up. The way we handle it on a research basis is that there's always a referring physician, often a geneticist, and we call and talk to that person and sort of together decide what to do about it. Sometimes the information is returned to the patient, and sometimes it has not been. And our consents currently are that we won't return them, but we're not always comfortable knowing that this person carries a BRCA1 mutation and not telling them that. So we sometimes do. This hasn't been sorted out. You know that, I'm sure you know that the American College of Medical Genetics and Genomics has put forward a policy statement that says that when a laboratory performs whole exome or whole genome sequencing, they must look for mutations of, I think it's 56 genes that are clinically actionable when that sequencing is done for any reason except prenatal diagnosis. So you may be looking for an intellectual disability gene in a 2-year-old child, but if you find a BRCA1 mutation, you have to tell the physician who ordered it. The physician who ordered it is then in a health of pickle if they haven't spoken to the patient about the possibility of incidental findings, which they probably haven't, unless it came through genetics and you have a compulsive genetic counselor. So you've got problems. That's how we handle it, but we're very uncomfortable with it. We're doing research in this area. I can tell you what we're trying to do is to develop a computerized method of pre-test genetic counseling.
That's all the time we have.
Okay.
So thank you very much.